Hello, welcome to my talk where I'll be sharing with you some of the medchem work we've been doing at the Institute of Cancer Research, generating cell-active inhibitors of BCL6, on the way to developing a therapeutic for diffuse large B-cell lymphomas. So I'll start with an introduction to BCL6 and why you might use it as a therapeutic target for these lymphomas before talking through our hit discovery strategy, a hit to lead process, which culminated in the nomination of an inhibitor for our lead optimization stage. I'll then go through some more recent progress and then have a look back at some conclusions. So why target BCL6? So BCL6 is a transcriptional repressor, which through binding to its co-repressor peptides, B core and N core at its BTB domain, represses certain genes. This is important in generating an immune response. For example, the BCL6 induced repression enables germinal center B cells to proliferate rapidly, evade growth checkpoint controls, and tolerate genomic instability, which is important obviously in affinity maturation in immune response, but it's also a process which is somewhat hijacked in cancer cells where the deregulation of BCL6 allows lymphoma cells to escape apoptosis, tolerate DNA damage, and you get this genetic heterogeneity amongst tumours. And most B-cell lymphomas arise from germinal center B cells and are key. And the key thing is they are dependent upon continued expression of BCL6 for survival. Our therapeutic hypothesis, therefore, is disrupting the binding of co-repressors to BCL6 would eliminate gene repression, and this would provide an effective treatment for BCL6-driven lymphoma. Looking in closer detail at the BTB domain and this binding site, the co-repressors bind along a channel formed by homodimerization of the two BCL6 monomers. So it's a unique binding site, so we weren't so worried about selectivity, but it is a protein-protein interaction, so it's shallow and it's polar, but it does have a calculated druggability score comparable to other successfully targeted protein-protein interactions. When we started the project way back when, there were actually very few available tool compounds. There were some peptides and some weakly binding small molecules such as 59,6. So our starting point was a broad range of hit finding experiments. So we did a virtual screen, a high throughput screen, and a fragment screen. And we identified multiple weak binders. For example, from the high throughput screen, we generated these two hits. We were able to develop multiple assay formats to enable the screening and the cross-validation of hits. These poorly soluble hits were optimized by synthesis and analog and enabled confirmation of activity by SPR, NMR, and X-ray. These two hits, looking a bit more detail and in the crystal structure, bound similarly. They had common interactions in their X-ray structures. They had a hydrogen bond to methionine 51 on the backbone carbonyl, and they also had this hydrophobic interaction with tyrosine 58. Merging these two hit compounds led to our first potency breakthrough, this compound 386. It now has a single-digit micromolar biochemical potency. So looking at this compound 386 in more detail, we can see it has a good ligand efficiency for a protein-protein interaction hit compound. It has high permeability and low clearance. In the crystal structure, we can see an extension to the pocket to the right of the compound, which is highly conserved amongst all of our crystal structures. In addition, in this pocket, there's a highly conserved network of water molecules, also conserved throughout all of our structures. The benzimidazolone compound has a single vector from this methyl group into this pocket. So at this stage in the project, we were wondering whether if we change the scaffold, we'd be given additional vectors into this pocket, for example, the quinolinones scaffold change. That's not to say the benzimidazolone series could not be optimized. It was, and from this series, we've generated inhibitors and degraders for BCL6. So our hypothesis at this stage was binding in the extended pocket would improve potency, and a scaffold change to a quinolinone would provide different vectors in order to do that. Our proof of concept compound, the quinolinone 388, was made and has comparable potency to the benzimidazolones. When we looked at the crystal structure of this compound, however, we saw that it bound in two different conformations, one in which the C4 of the quinolinone points out into solvent in a sort of solvent-facing orientation, or the C4 of the quinolinone points down into the pocket. As I mentioned before, the water molecules in this extended pocket are highly conserved across all of our crystal structures, but we want to know a bit more about them and see how we could target them in order to increase our potency. So we conducted a SHMAP analysis, which is a computational tool whereby you calculate the relative free energy of a charged probe or an uncharged probe at a specific location within a crystal structure. For example, we took the locations of the water molecules in turn. So the value it gives you shows you whether there's a preference for one of these probes over the other. For example, if you have a positive value, it suggests that an uncharged probe or a hydrophobic species would be energetically preferred, or if you have a negative value, it will show that a polar atom or a charged species would be more energetically favourable. So a hypothesis would be that substituent would bind reliably in the pocket, would anchor one of these conformations, and that substituent would also provide a vector to explore the water-filled pocket. And looking more in close detail, we can see that a ligand from the 4 position of the pocket-facing orientation could displace water 1 and give us new interactions to provide potency. To this end, we synthesized a set of quinolinones substitution in the 4 position. However, we saw flat SAR. There was no magnitude change in the potency that we saw across the board. We subjected these compounds to crystallography and looked specifically for whether there was ligand density in the extended pocket, and so therefore we could come up with an idea of whether there was solvent-facing or pocket-facing orientations. With the unsubstituted compounds, I've already shown you there were both orientations seen, and actually the ligand density for both was essentially equal, showing there was no preference for one over the other. The pyrazole compound here also bound in 2 different conformations, and making the pyrazole compound bigger just made it flip out into the solvent-facing orientation. And actually, over all these compounds, it was only the N-ethylamine compound here which bound solely in the pocket-facing orientation. So we took this through, we refined the X-ray structure of this ethylamine 003 compound. So we can see here that we've got the water 1 has been displaced, and there is a new hydrogen bond between the NH of the ethylamine to alanine 52. The methyl group is actually forming some more hydrophobic interactions in a pocket near valine 18. Our hypothesis at this stage was that we could displace further waters from vectors created from the ethylamine substitution, and that our SHMAP analysis, which we reconducted, would suggest that polar substituents would be more favourable. So what we did then was to add an amide group in this position, and that gave us a 3-fold improvement in potency. The X-ray of this racemic amide showed distinct binding modes for each enantiomer, showing that the methyl group was important for anchoring the ligand in one conformation over another. We resolved the racemic mixture into its constituent enantiomers, and it sort of gave us the expected result. The S enantiomer, the solvent-facing enantiomer, was a lot less potent than the R enantiomer. The R enantiomer itself gave us a 10-fold improvement in potency. You can see that the alpha-methyl group occupies that same hydrophobic pocket near valine 18 and displaces water 1. In addition, the amide displaces water 3 and has made 2 new hydrogen bond interactions with waters 2 and waters 4. So this was a really nice result. We've got a 10-fold improvement in potency. However, we've got low permeability, and actually this compound showed very poor cellular activity. So at this stage, we needed to improve permeability and hopefully improve the potency further. So the first thing we did was make the single change of adding a methyl group into the N1 position on the quinolinone. This, however, didn't lead to an improvement in our permeability in the first instance, but we sort of kept it there throughout the rest of the project to reduce liability later on. We removed a hydrogen bond donor which gave us improved permeability, but we lost the potency. We then decided to look at 5- and 6-membered heterocycles with the aim of maintaining hydrogen bond interactions in the same way as the amide, in the same positions as the amide. And this led us to the pyrimidine, which not only had improved potency, it had some permeability, and it gave us our first micromolar cellular activity. The X-ray of this shows that the alpha methyl group occupies the hydrophobic pocket, and we've displaced water 1. In addition, we've displaced water 3, and we've got 2 hydrogen bond interactions with water 2 and waters 4. As with the amides, the single enantiomer gave an expected improvement in potency, but there was still scope to improve the permeability further. The first way that we tried to do this was by the addition of a cyclopropyl group into the para position of the pyrimidine, and this brought us a 2-fold improvement in potency and increased our permeability. The second thing we did was add an additional methyl group in the alpha position, and that also brought us a 2-fold potency improvement. The X-ray shows that it maintains the interactions of the monomethyl analog, but in addition, the second water molecule displaces water 5 right out of the back of the pocket. And nicely, this removes the stereogenic center in the molecule, which sort of makes it easier for further studies. So these 2 changes actually quite nicely combined together led to a piece of additive SAR. So we have this compound 347, which was now our lead compound for the project. It has high permeability and sub-micromolar cellular potency, in addition to its 26 nanomolar and biochemical IC50. So this compound in more detail, we can sort of see that we've displaced waters 1, 3 and 5. We've got hydrogen bonds to waters 2 and waters 4, and quite nicely we can see the cyclopropyl group folds down the edge of the pocket. It showed optimized chemical binding and good physiochemical properties, including high permeability, low clearance. It showed submicromolar cell viability inhibition in our SGDHL4 cells also, and at this stage it was nominated to be our lead inhibitor for BCL6. So as a sort of summary of where we came from, so we started with these 2 hit compounds for our initial high throughput screen. We've got a greater than 4,000-fold improvement in the potency, and this compound 347 and similar compounds showed overall acceptable properties to progress to lead optimization. We've continued to work on this series and have generated compounds with additional improvements in biochemical and cellular activity. A summary of what happened to the waters, basically. So 5 out of the 6 of the waters have been interacted with or displaced, which is sort of demonstrated quite nicely here. So in conclusion then, we started the project with a broad hit finding strategy. We had the most productive approach from the high throughput screen, even though it yielded fragment-like hit matter. We got our first potency hike through structure-based merging of those 2 hits. And then our strategy to pick up new interactions in the extended pocket started with rescaffolding to generate new vectors. We needed to conformationally anchor the quinolinone series, and then we used water analysis to design new substituents. We were able to achieve this potency breakthrough through replacing or interacting with 5 out of the 6 conserved water molecules, and we got a compound 347 as a compound to enter lead optimization phase. Further optimization of 347 has led to significantly more potent compounds. And then I'd just like to end on our acknowledgement slide. So Swen Hoelder, who's the project lead and the chemistry lead, and Olivia Rossinides, who is the biology leader, and then the rest of all the different teams, which make up the BCL6 project team. And I'd like to thank you for your attention. I'll take questions next.